hope it's visible. Is my screen visible? Okay. Thank you for the response. So I'll start now. So this is week 4 of the sample problems. The subject is applied environmental microbiology. So I am TA for this course. My name is Manchi Tripathi. I am PMRF research scholar from the Department of Civil Engineering. I am from the Environmental Engineering Division of IIT Madras. So how we will go on with this? There will be few concepts. Apart from that, there will be questions that you guys need to answer. So the questions are the representative of the questions that you will see in the assignment as well as in the exams. So this week 4 was about the microbial ecosystem. So we learned the difference between evolution, succession, then a population, community, and guilds. Apart from that, we studied about quantification. Here we studied richness, evenness, and other things. Also, you learnt about various indexes. Those were Shannon index and other indexes. So the first thing is what, if you remember, what is the difference between the evolution and succession? Other than that, what is the difference between population and community? Is there anyone who can answer this? You can respond also, or you can type in the chat box. First, the difference between evolution and succession. You can type in the box. So what is evolution? Okay, so here is the hint. In what of in which of these terms there is change in the genetic markup? Or where there is change in a complete species change? Yes, correct. Yes, succession is a gradual change. Yeah, but what happens in this thing? So what changes? Okay, both of these terms are related to the changes in the species with time. But when we talk about evolution, there is change in the species, there is change in the genetic markup. Like what will happen if if the temperature, for example, if the temperature for now is 20 degrees Celsius, so there will be certain level of certain type of bacteria with certain level of temperature accommodation. Okay, but when this temperature will change to 30 degrees Celsius, some of the bacteria will die which cannot not at all survive, but with time there will be change in their genes which will help them to survive in higher temperature. So this is evolution. But what happens in the other term is there is complete species change. Okay, there will be complete change in the species. So this also happens with respect to time. And what is the difference between population and community? What is the difference between population, community, or consortia? Yes, population is same group, same species group. And what is community? Okay, in which terms there is an interdependency? There are some cases in which two different type of species are there, but they are interdependent, like algal bacteria consortia. But there are some cases in which species coexist, but they do not cause anything, like either antagonistic or helpful. There is no such interaction between them. Yes, communities, one or more species live closely. So, is there any interdependence in between them in this community? Are they independent or interdependent? And what is consortia? Yes, correct. Okay, so I just wanted to revise these terms because these terms will be used throughout the course. So the next thing that we studied were indices. So first was Simpson index. It measures both richness and proportion. And I've given the formula also. Next was Shannon Wiener index. So it gives more importance to evenness. It it measures richness, but more importance is given to evenness. So mostly these two types of index are used, Simpson and Shannon. So whenever you want to do any community analysis or microbial identification, when the samples are given to the team to determine what kind of microbes are there, mostly the answer that comes is in the form of indices. So this is one example in which OTU is measured. We can measure abundance, then PI, LNPI. All these terms are there. It has been discussed in the lecture, but we will discuss it further. So this is just for an example that how to measure all these indices. Simpson index is abbreviated as D and Shannon as H. So the most important thing is to for any species, not only microbes or any macro species, humans and animal, two things are required. First thing is resource, and the second thing is right condition. Even if there is so much of resource, so much of food, everything is available, if the conditions are not right, what if the temperature is like 100 degrees Celsius? No human can survive, right? Even if there are resources. So similarly, if there is spring season everywhere, very good environment, but no resources, no water, then also no one can survive. So like that, for survival, two things are very important: resources and right condition. When we talk about resources, there are different type of organisms which feeds on different things. Some needs inorganic carbon, some organic carbon. Like for us humans, we need organic carbon. Heterotrophic bacteria, they needs organic carbon, but the autotrophic one or the trees, they need inorganic carbons in the form of carbon dioxide or other sources. Secondly, it's nitrogen. So it is also used in the form of organic or inorganic sources. When we talk about fermentation and all, the nitrogen and other nutrient sources are inorganic. However, when we talk about aerobic condition, it's organic. Other micronutrients are also required, like sulfur, phosphorus, potassium, magnesium, other things. Apart from this, there are some bacteria you might have heard about, diatoms and all, they need silica. Next important thing is electron acceptors, and then inorganic electron donors. So these are the resources that are required. These resources are required in one or another form for different type of microbes to survive. The other important thing is right condition. So we can talk about temperature, pH, ORP, light, and osmotic condition. So temperature I already explained. There are some bacteria which will die in higher temperature, but the thermophilic bacteria, they can survive in more than 60 degrees Celsius. Similarly, these pH bacteria, acidophiles, all these bacteria you might have heard about, Acidophiles live in acidic environment, like acido plus philes. Philes means loving and acido means acidic. So they will love the acidic environment, but they cannot survive in the basic environment. Next is oxidation reduction potential. So if the ORP is positive, aerobic bacteria can survive. If it is negative, the anaerobic bacteria can survive, but they cannot survive in the opposite condition. Light is also very important. Some bacteria can survive in dark, but some do need light, like cyanobacteria. The next thing is osmotic condition. Osmotic condition can be also attributed to the salt condition. Like if they are harophiles, it means they can survive in high saline condition. So even so, the osmotic condition depends upon the salts present in the environment. So this was the slide which was I tried to help you in identifying the resources and the right conditions. So these are examples of various resources and different conditions which will help the microbes to survive. One more important thing that we discussed in this lecture in this week was the surface attachment. So we know that microbes are mostly mobile in nature. But when there is lack of nutrients, they try to attach themselves to a surface. So when there is lack of nutrients, what they will do, instead of being mobile, they will release some EPS, that is extracellular polymeric substance. They will release that and it will help them attach to each other or to the surface. When they are in this attachment position, they will use less resources. So what happens, like in lake, there is more nutrients compared in comparison to river. So in river is in flowy condition. So what happens, the bacteria will try to attach itself to the rock or something. So the rocks nearby river or any water body is mostly slimy. If you have noticed, it's slimy, sometimes it's green, so algae also attaches itself. All these bacteria, algae and other microbes, they tend to float. But in case of low nutrients availability, low light availability, and any condition which is harsh for them, they agglomerate and they attach themselves to the walls or to the side of rocks and all. So this helps. In this way, they are not using their body parts much. So what happens, they will use all of their energy in capturing nutrients, and they can save the inner cells. So when they agglomerate, the size increases. So how the size increases, it's not the single bacteria whose size is increasing. The outside bacteria, it's coming and attaching itself to the sides, and they are trying to save the inner bacteria. So in the process of saving the inner bacteria, the outer bacteria goes into sacrificial mode. So they started dying and sloughing off. Okay, so this is how this self attachment or surface attachment saves bacteria from dying. So here the outer bacteria goes into sacrificial mode and one can save the inner bacteria. How do they do this? They form a biofilm which can secrete extracellular polymeric substance, that is EPS in short. So it forms a mat over the surface, that slimy thing that you see. It forms a mat and then it captures the nutrients from the flowing water. So this is how it survives. We also studied about aquatic ecosystem. So there is atmospheric water which is present like in form of condensation when we sweat or something, atmospheric water. Second is surface water and the groundwater. So for public health, surface and groundwater health is important. So like our major source of water is surface or groundwater. There are technologies which can capture atmospheric water. You can search, there is a concept, a program, Jalvayu. So that is a machine that captures water from the atmosphere and converts it into drinking water. So that machine is called Jal Vayu. It is patented technology. You can look for that in Google, but mostly this is high class technology. So mostly the technology, the water that one use is surface and the groundwater. Okay. So whenever we talk about the microbial ecosystem in drinking water, we mostly talk about surface and groundwater. So for measuring the microbial contamination, the most important parameter is BOD, that is biochemical oxygen demand. So this is used for the quality control in wastewater treatment plant and water treatment plant. It is also a effluent standard. What is effluent standard? Like we have wastewater, we want to treat it, so we will take it to wastewater treatment plant. So whatever the wastewater that goes out of our houses and industries and other places, so let's talk about house first. Like after bathing, brushing, flushing, everything, the water goes down in the drain. So these wastewater treatment plants are there which are used for treating this dirty water. So the water that goes inside this wastewater treatment plant is called influent, influent, influent. And after treatment, the water comes out of the wastewater treatment plant, so that is called effluent. So whatever is the concentration of the compounds, harmful compounds in the water, in the influent, the effluent will have the effluent will be relatively clean. So the mostly we will talk about BOD values. So what happens, this BOD, this biochemical oxygen demand, the value of the BOD should reduce. So there is certain level of BOD which is there when we want to discharge this water into any water body. So mostly is 20 milligram per liter. So there is always a standard, like we cannot just discharge all the dirty water inside the water body, right? We clean it up to a certain level, and once that level is reached, we discharge it into the water. So that level is called effluent standard. And what is this ambient standard? Ambient standard means the receiving water body, like we are discharging dirty water into the lake or river. So what is the level of BOD in that lake or river? So that is called ambient standards. So effluent standard is whatever is coming out of the water treatment plant is effluent standard, and ambient is whatever is already existing in the water body is called ambient standards. So for maintaining this ambient and effluent standards, there are various rules and regulations set by the government, and which is being prescribed by the local bodies, urban local bodies. So the value of 20 milligram per liter, whatever I am saying, is effluent standards. It is been decided by after doing various research, and it has been followed by government. Next thing that was discussed in the lecture was dissolved oxygen, that is DO problem. So the problem of dissolved oxygen in surface water has been recognized for over a century. We know that, right? So when we want to know how good the water is, how good the ambient water is, like lake or reservoir or river, we measure the dissolved oxygen. If the dissolved oxygen is more than a certain level, let's say 4 milligrams per liter, we know that the fishes and other aquatic organisms, they will survive properly. When the oxygen level goes below a certain level, let's say 4 milligrams per liter, the aquatic organisms, the fishes, they won't be able to survive. So we say that the water is sick. Okay, water is not fit. So this is why dissolved oxygen measurement is very important. So DO is a surrogate variable for the general health of the aquatic ecosystem. This I have already told. DO present in surface water is often used as index of water quality. The impact of low DO concentration or of anaerobic condition was reflected in an unbalanced system. So either the DO is low DO means less than 4 ppm, 4 milligrams per liter, or anaerobic condition. What is anaerobic condition? No DO at all, completely lack of dissolved oxygen. Unbalanced system. So it represents what? Fish mortality. Fish mortality means that I told you, if the DO is less than 4 milligrams per liter, the aquatic organisms won't be able to survive. So they will die off. So that is called mortality. Next thing is foul odor and aesthetic nuisance. When these fishes and other aquatic organisms will die, there won't be enough bacteria, because bacteria also survive on dissolved oxygen. So these things will start, the fermentation process will start in the river or any water body due to anaerobic condition. So when there is anaerobic condition, the foul smell will start coming out of methane, H2S. H2S means hydrogen disulfide, and methane, CH4. So the smell will start coming out, and the water will become dark. So it will not look good to the eye. So there are aesthetic issue. So the DO problem can be summarized as the problems of organic and inorganic oxidizable residues into the water body, which during the process of ultimate stabilization of oxidizable material in water or sediments and through interaction of aquatic plant life results in the decrease of DO to concentration that interfere with decidable water uses. So what happens? Due to excessive discharge of organic and inorganic waste into the water body. So these organic and inorganic waste, like if I discharge a plastic, so it is though organic, it is polyethylene, it is organic, but what happens? It will take a very long time to degrade. So it is not going to consume DO. But if I am discharging any waste from the fields or from the cattle field, all those things, from any sugarcane industry, so when they are discharged into the water body, they will, for deteriorating them, the bacteria will start consuming oxygen, so it will reduce the oxygen. Okay, so that is dissolved oxygen problem. Next, that I was explaining earlier, biochemical oxygen demand, that is BOD. So it is defined as the amount of oxygen consumed during microbial utilization of organics. Okay, so amount of oxygen consumed during microbial utilization of organics. So here the microbes that are there are heterotrophic bacteria, not autotrophic. Heterotrophic bacteria utilize organics, autotrophic utilize inorganics. So that is why the bacteria which are present here, this question was there in the last semester. So the amount of oxygen consumed during microbial or heterotrophic microbial utilization of organics is called biochemical oxygen demand, or BOD. So the BOD is measured by determining the oxygen consumed from the sample placed in an airtight container and kept in a controlled environment for a pre-selected period of time. So how do we perform this experiment? We take 300 ml BOD bottles. Okay, 300 ml BOD bottles. We keep the known amount of sample, like 5 ml, 10 ml. The sample is the one for which we want to determine the BOD. 5 ml, 10 ml, like that we keep the samples. Then we keep we fill the bottle up to the brim so that we can make it airtight using aerated water, aerated good quality water. Okay, we use distilled water and we aerate it completely so that the DO level reaches saturation, and we fill the 300 ml bottle with that, with the sample inside. Then we close the bottles to make it airtight. Then we can determine the BOD for 3 days or 5 days depending upon the resources we have and the time. Time and resources are very important for measuring the BOD. If we have less time, then we can measure for 3 days. If we have enough time, then we should measure for 5 days. So for 5 days we keep this sample in 20 degrees Celsius. Okay, and if we have less time, then we keep it for 3 days at 27 degrees Celsius. So that is very important. 5 days 20 degrees Celsius and 7 days 27 degrees 3 days 27 degrees Celsius. Okay, other most important thing is that light must not be included in the incubator. These bottles should be kept in dark. Why? Because if there is light, we know in the samples there are chances of algal cell as well. So if there are algal cell, they will find out light. If they get any light, they will start growing. So when they will start growing, they will start producing the oxygen. So we will not get the realistic BOD value, because what we are doing, we are measuring oxygen in the initial level and after 5 or 3 days depending upon the time we have. Okay, so because the saturation concentration of oxygen in water at 20 degrees Celsius is approximately 9 milligram per liter, dilution of the sample with BOD free oxygen saturated water is necessary to measure BOD values greater than just a few milligrams per liter. So as I've already told, we need to fill the bottles with BOD free saturated DO saturated water. So how does this biochemical oxygen demand helps us? So it first helps us to determine the oxygen consumption. It also tells us about the number of microorganisms. Further, it can be used to determine the amount of organics. So what is BOD? It is oxygen equivalent of organics. Okay, it is nothing but oxygen equivalent of organics. So let's get back to our questions. So this was the summary for the entire week. So our first question is, as we have seen in the table, first table that we have seen, first is OTU in collector's curve stand for. You can write the answer in the chat box. I'm waiting for more responses. Yes, okay. So the answer is operational taxonomic unit. So all of you are correct. So the next question is, evenness E is the measure of how similar the abundance of different species are if the community is as diverse and even as possible. So community is diverse and even. It is evenly diverse. So what will be the value of E, evenness? Okay. We'll see the answer. So E is 1. So yes, option A is correct. The next question is, which of the following conditions need to be favorable for the microbial community to thrive? We need to select the best option. So we studied about all these things, right? Right condition and resources. So which of the following is the most right condition? Yes, correct. All these conditions needs to be maintained in order for a microbe to survive. So this is a match the following type question. Please take your time to answer this question. We'll see the solution in a while. I need more responses. Give it a try. Simple. Okay. Sorry. So here is the solution. It is option B. So population is when we talk about same species. Okay. Community is when there are different species together. When we say guild, it can be a trait, a particular, when we talk about a particular trait, we call it guild. So one of the trait is sulfur reducing bacteria. So this is trait of many different species of bacteria. So it is a guild. What is algal bloom? It is high nitrogen and phosphorus level. We already know that high nitrogen and phosphorus level causes algal bloom. What is anaerobic sulfur oxidation? It is the property of purple phototrophic bacteria. And sulfur reduction, it is the property of desulfobryoi bacteria. Answer of second question. Answer of second question is option A. E is 1. So if the community is as diverse and even as possible, then E evenness index will come as 1. So coming back to question number 5. Microbes may form biofilm when they attach to the surface. Which of the following is true? What happens when they form a biofilm? Option A and option B are 50-50. So the correct option is option B, that is A, B and C. So biofilm gives strength to the grip on the surface. Yes, because it forms EPS layer. Biofilm provides protection. Yes, it provides protection to the inner bacteria and it sacrifices the outer bacteria. And biofilm captures nutrients. Okay, so C option is also correct. And option D is biofilm is the powerhouse of microbes. No, it is not. It does not provide any energy or anything. It helps to capture nutrients. It helps to adsorb nutrient because it is the PPS layer has sticky surface. So next question is, the microbial functional community structure changes with depth in an aquatic system due to, in an aquatic system like in ocean or a sea or any river, there is change in the microbial community structure from the top to the bottom. So why this happens? Is there any change in light or ORP, energy requirement, and or changes in cell composition? So what can be the correct reason for this? I'll give you a couple of minutes to answer this. So the answer is change in light intensity and reduction in oxidation potential. Okay, so the sunlight can penetrate up to a certain level. So when there is change in the light intensity, the bacteria which requires less light, which can survive in less light, lives in the bottom, and which can survive in more light goes to the top. Similarly, with depth, there is change in oxidation reduction potential because the light is less light is coming to the bottom. So what will happen? The plants which uses oxygen and light to produce which uses light to produce oxygen will be there more at the top surface. So oxygen is there at the top surface, but at the bottom layer there can be an anaerobic pockets. So what is happening? There is change in oxidation reduction potential. So mostly facultative or anaerobic bacteria will be there at the bottom, and aerobic bacteria will be there at the top. Therefore, there is change. So why this change in happening? Aerobic to facultative to anaerobic bacteria, because there is change in oxidation reduction potential. So these are the two reason there is change in microbial functional community, light and ORP. So energy requirement is not the reason. Okay, and there won't be any changes in the cell composition because the different bacteria are there will be changes in cell composition, but not the other way around. What are the two parameters that influences Simpson's index D? This also has been discussed, Shannon and Simpson index. So the answer is evenness and richness. Organomercury lyase converts. So this is when we say ASE, like anything which ends with ASE is enzyme. So organomercury lyase, which is an enzyme, converts methyl Hg, that is mercury, so methyl mercury, to methane and mercury. And the gene encoded to carry this out are what genes are there which are required to carry out this operation? Methyl mercury to methane and mercury. Yes, correct option is option D, that is MERA. Sulfur oxidation occurs. Select the best option. Answer is both anaerobically and aerobically. The next question is, nitrogen in both organic and inorganic form is essential for microbes. It is used in what process? Formation of the structure of DNA, or fixing nitrogen in the environment, or to carry out redox reduction, or to maintain the pH in the surrounding. Yes, all of you are correct. So this is used for the formation of structure of DNA, fixing nitrogen in the environment, and carry out redox reduction. I would like to mention very important thing in such kind of question. If you are marking only one answer is correct, then you won't get the marks. You need to mark all of them, which if there are many correct answer, you should mark all of them as correct. Then only you will get the marks. Okay, so for such kind of question, please look at all the options carefully, then mark the answer. So this is fill in the blanks kind of question without any options, very simple. So ecosystem combines dash and dash factors of the environment. What can be the answer? Yes, correct. Biotic and abiotic. So next is this question. Match the following. Give me the answer. Similar kind of question was done earlier. Yes, true, all are correct. So next question is, number of different kinds of organisms or types of species in a particular area are called. Different kinds of organisms are there in a particular area. What are they called? So answer is richness. We are talking about number. So we are talking about the quantification. Okay, read the question very carefully. It's not, I'm not talking about the definition. Here we are talking about the number of different kinds of bacteria. So that is why answer will be richness. Next question, evenness is a measure of the dash of different species making up the richness of an area. Yes, correct answer is relative abundance. Now we need to calculate the Simpson index of diversity. So this is OTU in abundance. Please take some time in calculation. We will see the answer in a while. So this is the solution. We have the OTU abundance. We need to calculate the PI, then PI square, then 1 minus T. Okay, everything is written here. So the correct answer is 0.844. Please match it with your own solution. How this PI is calculated? First we will add all the abundance values that came out to be 400. Now how this PI is calculated? That is for the first one it will be 45 divided by 400, then 50 divided by 400. Like that we will calculate the PI. Then we will calculate the PI square. This is simply like for the first one PI square is 0.113 multiplied by 0.113. So the next is sum of squares and we calculated D. I think the mistake that you have done is you have calculated D, but Simpson value is 1 minus D. Okay, so whatever you have calculated is D value. So that is the probability that the two randomly selected individuals in the community belong to the same category. But what is 1 minus D? It is the probability that two randomly selected individuals in a community belong to different categories. So what we needed to calculate was the different category, not the same category. So please note it down. Okay. So now we need to calculate the Shannon-Weiner index. Please solve this one. And please be careful, the abundance values are different from the previous one. They are not same, okay? So you need to make a new table. You can use Excel for solving it fast, or else you can do it manually. So this is question number 16. Calculate Shannon-Weiner index. I'll give you 5 minutes to do so. Sorry. Yeah. So this is the solution. Similarly, we will calculate the pi, then we will calculate natural log of pi. Then we will just multiply pi with natural log of pi, and the answer is 1.880. So yes, your answer is correct, Mary. Other people, please note it down. So we'll move forward to the next one. Okay, so the question number 17 is a very simple one. Which of the following statement about redox potential is false? NAD plus NADH redox pair has the highest redox potential, or oxygen water? The components of ETC transport chain are organized in terms of their redox potential, or the redox potential of the system is usually compared with the potential of hydrogen electrode. Which, are, which among the following is false?
是封面这样的。
So the wrong option here is NAD plus NADH redox pair has the highest redox potential. Okay, other options are correct. So which of the following is non-photosynthetic bacteria? Or which of the following is non-photosynthetic microorganisms? Not all of them are bacteria. Correct. So the next match of the following type question is there. We need to match the following with respect to terrestrial environment. Previous question is option D only. So Anthony, the previous question is option D. The others are photosynthetic. Only the last one is non-photosynthetic. So the next question is match the following with respect to terrestrial environment. Other students, please answer. Okay. So all of your answers are correct. Okay. And this is the last question. ORP determines the functional class of microbes in the environment. True or false? This has been previously discussed as well. So this is the statement. Okay, very correct. So the answer is true. So that's all for today. So I'll still keep my I'll keep myself online for a while. So in case of doubt, you can type in the chat box. I would love to answer. And other people, you all can leave. I will keep myself online for 10-15 minutes. So other people can leave. So that's all for today. Thank you everyone. Which question, Sayanthani? Sayanti, I'm sorry. Which question do you want to know? Question number 20. The answer is true. So we were discussing this, right? ORP, the functional the resources and the right conditions. The right condition was pH, ORP, light. So this is one of them. So ORP determines the functional class of microbes because the ORP will change, right? At the top surface it will be aerobic, and at the bottom it will become first facultative, then it will become anaerobic. So ORP is changing. Thank you everyone.
किसकी जरूरत नहीं तो ठीक है लेक्चर हो गया ना अब मतलब बैटरी बची है और अपलोड करना है अपलोड के बाद 1.5 घंटे का ही करना होता है ना 7 से 9 होता है 7 से 9 होता है तो 8:30 अभी 12 मिनट होता था 12 मिनट और बचा होगा मैं नूडल्स बना रहा हूँ दो चटनी निकालना यहाँ हाथ से
ये तो अच्छा है, ये तो खा सकते हैं।
सब अच्छी क्या बता दी बता दी
डायरेक्ट बन जाता है, इसमें थोड़ा सा
ये उस दिन वाला तो नहीं है ना पेट्रोल वाला यूज किया था स्वागत नहीं है ट्रेन
कि पानी का मार्क नहीं बनेगा, इसको स्ट्रोक करना होता है। ये थोड़ा सा अलग ब्रांड है।
हाथ से कर लो।
हाथ में नाखून साफ है, साफ रहना नहीं, ऑयल लग जाएगा तो बहुत।
मैं कर दूँ। एक बार आज पहले दिन तो मैंने पानी जाम चाहिए। तुमको अच्छा लगता है तो जाम हाँ हाँ अभी पता चला अभी हुआ ना बाजार अरे सब ठीक है कुक कर रहे हैं डिनर कपड़े पहन के फॉर्चुनेटली नहीं है क्या अपनी बॉटल्स खत्म हो गई सारी जो बैग में थी वो क्या मैं सर्व करूँ खोलोगी मैं पिलाता हूँ so this bottle yes some water for my baby क्या एकदम प्रोफेशनल फील कर ये वो बोतल है जो बोला जाता है कि खत्म करनी है, बॉटम्स अप। पैर में लग जाएगी, इसको उठा लो। और क्या तो है, यही जलाएंगे, बाकी सब ऑफ कर देंगे।
अभी भी 4 लोग आराम से बचे हैं। अब बैटरी मेरी रोना स्टार्ट शुरू कर चुकी है।
ये आज ही के दिन अपलोड भी करना होता है क्या? अपलोड बाद में
मैं कल कर सकती हूँ, एक दिन रुक सकते हैं।
एक दिन क्या करता, 2-3 दिन रुकना पड़ेगा।
हाँ, क्योंकि अब मेरी बैटरी कितनी बची है और अभी 3 मिनट बचा हुआ है। बैटरी 25% है।
कितना?
3 मिनट अभी भी बचा हुआ है।
कितना परसेंट है?
25। चल जाएगा, चल जाएगा।
ब्राइटनेस मिनिमम कर दो ना।
ब्राइटनेस मिनिमम है।
तो छेड़खानी मत करो,
मिनिमम ही है।
तो इधर ऐसे आइडल रहने दो सर।
रहो बेटा, आइडल रहो, आइडल रहो, आइडल रहो।
3 मिनट बचा है ना, बस तो हो जाएगा। ये चार्जर में पता नहीं कोई काम आएगा। छोड़ दो फिर गुलती रहोगी 3 मिनट।
2 मिनट।
चीज़ें बस मैं खिलाना नहीं चाह रहा हूँ, इधर उधर खेलो।
Dun dun dun.